What if everything you thought you knew about the people who built the pyramids was written in a code so small, so fragile, that scientists spent decades believing it could never survive the desert at all? For years, the world's leading geneticists assumed that the scorching heat of Egypt had erased the genetic memory of one of history's greatest civilizations forever. DNA, they knew, breaks so, down in heat. It fractures, it degrades, it vanishes into dust, just like everything else time touches. And yet, inside the sealed chambers of an ancient burial site along the Nile, something impossible had been waiting. Something that would force scientists to ask a question few had dared to ask before. Were the ancient Egyptians, who built one of history's most dazzling civilizations, genetically the same people who walk the streets of modern Cairo today? The answer, when it finally came, stunned even the researchers who found it. To understand why this discovery mattered so much, you first have to understand just how deep inside us this story is written. Every human cell carries roughly 2 meters of tightly coiled genetic thread, a molecule called deoxyribonucleic acid, DNA, twisted into the elegant double helix, first described by James Watson and Francis Crick in 1953, built on the overlooked but essential work of Rosalind Franklin, whose X-ray images captured the shape of life itself before anyone truly understood what they were looking at. Inside that helix are 4 chemical letters: adenine, thymine, guanine, and cytosine, arranged in sequences that spell out the instructions for building a human being, or a wheat plant, or a Nile crocodile. Change the order of those letters even slightly, and you change eye color, disease risk, ancestry, sometimes the very course of a life. It is, quite literally, the most information-dense material known to science, capable of storing the entire blueprint of a person in something too small to see without a microscope. But DNA is not indestructible. It is, in fact, remarkably delicate. Over time, exposed to heat, moisture, and microbial invasion, the long strands of genetic code shatter into smaller and smaller fragments, like an ancient scroll left in the rain until the ink runs and the parchment crumbles. This is why, for a long time, ancient DNA research focused almost entirely on cold environments. Siberian permafrost, Arctic ice cores, alpine glaciers. Scientists extracted stunning genomes from tens of thousands of years ago in these frozen archives, piecing together the migration of early humans and the shadowy genetic ghosts of extinct cousins like the Neanderthals and the Denisovans. Egypt, by comparison, seemed hopeless. Average summer temperatures regularly exceeding 40 degrees Celsius. Millennia of heat, cycling through tombs and burial pits. Every textbook on ancient DNA preservation essentially agreed. Egyptian mummies were a genetic dead end. And then a team of researchers, led by geneticists Johannes Krause and Verena Schunemann, working with colleagues across Germany and the United Kingdom, decided to test that assumption anyway. Their target was a site called Abu Sir el-Malek, a settlement and burial ground along the Nile roughly 115 kilometers south of Cairo, occupied for well over 1,000 years. Hundreds of mummies had been excavated there over the past century, spanning a staggering window of time from around 1400 BCE, deep in the New Kingdom era of pharaohs and temples all the way, through the Ptolemaic period after Alexander the Great's conquest, and into Roman rule nearly 400 CE. If ancient Egyptian DNA could be recovered anywhere, this sprawling, well-documented necropolis was one of the best chances science had ever had. It's worth pausing here to marvel at just how strange and beautiful the science of reading this history actually is. A mutation is, at its core, nothing more than a copying error, a tiny mistake made when a cell divides and DNA replicates itself. Most mutations are meaningless, invisible, absorbed into the vast 3 billion letter code without consequence. But every so often, a mutation survives, gets passed down, and spreads through a population, sometimes because it offers some subtle advantage. Resistance to a disease, tolerance for a new food source, sometimes for no reason at all beyond pure chance. Geneticists call this genetic drift, and over enough generations, these small, silent copying errors become the very fingerprints scientists use to trace where a population came from, who they mixed with, and when. It means that somewhere in the DNA of every living person is a kind of accidental diary, written not in ink, but in the statistical noise of billions of cell divisions stretching back to the dawn of our species. The same technology, the ability to extract, sequence, and compare ancient genetic material, has already rewritten human history once before, and it did so in a way that still unsettles people today. In 2010, geneticist Svante Pääbo and his team announced the sequencing of the Neanderthal genome, drawn from bone fragments found in a cave in Croatia. The discovery revealed something almost nobody expected, that Neanderthals were not simply an extinct evolutionary dead end, wiped out and replaced without a trace. They had interbred with the ancestors of modern humans migrating out of Africa. Today, most people of non-African descent carry somewhere between 1 and 2% Neanderthal DNA, fragments of an entirely different species of human, embedded quietly in the code of billions of people alive right now, most of whom have absolutely no idea it's there. Pabo would go on to win the Nobel Prize in Physiology or Medicine in 2022 for this body of work, a recognition of just how profoundly ancient genetics had reshaped our understanding of what it means to be human. It's a humbling thought. The boundary between species that once seemed absolute turns out to be far blurrier, far more intimate than anyone imagined. Egypt offers its own version of this kind of humbling discovery, and it isn't limited to the Abusir El-Malek study alone. Perhaps the most famous and most contested attempt to read royal Egyptian DNA came in 2010, when Egyptian antiquities authorities, led by Zahi Hawass, working with an international team of radiologists and geneticists, examined the mummy of Tutankhamun and 10 other royaux, mummies believed to be members of his family. Using DNA analysis alongside CT imaging, the team proposed a 5-generation family tree, tentatively identifying Tutankhamun's father as the mummy known as KV55, widely believed to be the pharaoh Akhenaten, and suggested that Tutankhamun's parents were, in fact, siblings, a practice not uncommon among Egyptian royalty seeking to preserve bloodline purity and divine legitimacy. The study also detected genetic evidence consistent with multiple strains of malaria in Tutankhamun's remains, along with signs of bone necrosis, and linen wrapping presents unique preservation and contamination challenges distinct from the natural desiccation seen in more modest burials like those at Abusir El-Malek. And some geneticists have since questioned aspects of the methodology, and Egypt offers its own version of this kind of humbling discovery, and it isn't limited to the Abusir El-Malek study alone. Perhaps the most famous and most contested attempt to read royal Egyptian DNA came in 2010, when Egyptian antiquities authorities, led by Zahi Hawass, working with an international team of radiologists and geneticists, examined the mummy of Tutankhamun and 10 other royal mummies believed to be members of his family. Using DNA analysis alongside CT imaging, the team proposed a 5-generation family tree, tentatively identifying Tutankhamun's father as the mummy known as KV55, widely believed to be the pharaoh Akhenaten, and suggested that Tutankhamun's parents were, in fact, siblings, a practice not uncommon among Egyptian royalty seeking to preserve bloodline purity and divine legitimacy. The study also detected genetic evidence consistent with multiple strains of malaria in Tutankhamun's remains, along with signs of bone necrosis, offering a possible, though still debated, explanation for the health struggles that plagued the young pharaoh before his death around the age of 19. This study sparked significant scientific debate. Ancient DNA from royal mummies, preserved through artificial embalming with resins, natron salt, and linen wrapping, presents unique preservation and contamination challenges, distinct from the natural desiccation seen in more modest burials, like those at Abusir El-Malek, and some geneticists have since questioned aspects of the methodology and the certainty of the familial conclusions drawn. It remains a striking, if still contested, example of how far this science had already come and how much careful scrutiny each new claim requires before it can be trusted. What both of these Egyptian studies reveal together is years of work and roughly $3 billion in explosive advancement in sequencing technology that finally made deep, reliable extraction from fragile, heat-damaged ancient Egyptian remains achievable at all. This raises an irresistible question, one the researchers themselves have had to sit with. If scientists can now read the deep genetic history hidden inside 3,000 years, old mummies, what else might be waiting undiscovered across the rest of Egypt and across the rest of the ancient world? The Abusir El-Malek study examined mummies from a single necropolis along the Nile's middle stretch. Egypt's history spans more than 3,000 years of dynastic rule and stretches across a geography touching Nubia, the Mediterranean, and the Sinai. Somewhere beneath the sand, in tombs not yet opened and collections not yet tested, may lie genetic answers to questions historians have debated for over a century. The true biological relationships within contested royal dynasties, the genetic footprint of foreign rulers like the Hyksos or the Ptolemaic Greeks, the deeper ancestral connections between Upper Egypt and the kingdoms of ancient Nubia and Kush to the south. Every mummy successfully sequenced doesn't just answer a question, it reveals how many more questions remain completely unanswered, and this science is no longer confined to museums and academic journals. It has quietly moved into the world of the living and rare inherited disorders long before symptoms ever appear. Genetic counselors can now tell prospective parents whether they carry recessive genes for conditions like cystic fibrosis or sickle cell disease. Forensic genetics, built on the very same principles used to authenticate ancient DNA against contamination, now helps identify victims of mass disasters, exonerate the wrongly convicted, and solve decades-old cold cases using genealogy databases that trace distant family connections through shared fragments of ancient inheritance.